institution for giving a platform to share our research findings at this occasion. Move on to the topic, key role of fused-dose homolog in cervical carcinogenesis. Cervical cancer, which arises in the lower part of the uterus, which connects to the vagina. The basic key role of human papilloma virus, which is involved in the cervical. But in some
Prabakaran sir, Dr.
Sir?
Prabakaran sir,
yes, sir.
how are you, sir?
Hello?
Can you hear my voice?
Yes, sir. Yes, sir. I can hear you.
Okay. Sorry, sir, unable to join on time, sir. Sorry, sir.
Okay, no problem. No problem. Shall I join?
Please, sir, please continue your session, please.
Okay. Thank you.
Thank you.
And what are the risk factors in developing cervical cancer and how to reduce that? There are two ways. One is the involves the collecting of cells from the cervix. And age of 30 should do the pap smear test in every 3 to 5 years routinely, and they recommended the HPV vaccines for the age of 12 to 15 years of women. What are the signs and symptoms? Signs and symptoms of cervical cancer. In the earlier stage, it doesn't show any symptoms, but in the more advanced stages, it shows the abnormal bleeding and pain during the menstruation, and the swelling of legs and a problem in urinating, and the blood which comes in the urine. There are different types of subtypes of HPV which are known. Among that, 40 can infect the epithelial lining. There are two types of HPV which is said to be the low risk HPV, high risk. Mostly the low risk which causes only the benign warts, and the high risk of HPV 16 and 18, which are the known cervical carcinogenesis. Also, the subtypes of HPV which are involved in vulva, vagina, and anal cancer in women and cancer in men. There are a set of oncogenes which are driven the progression of cervical cancer. Here, E5, E6, and E7, which are said to be the early genes, which play a key role in the initiation and progression of cervical cancer. And the other set of genes, E1 to E5, which are involved in the and here, HPV, which is a small circular double-stranded DNA, warts, but the high risk of HPV, which infect the basal epithelium of the cervix, and after several weeks, the viral replicate and hijacks the entire genome machinery, which leads to cervical cancer. And after several years, it leads to the invasive cancer. The molecular mechanism of how HPV infects and then if
Sir, there is an issue in
the
network.
large hello.
Sir, we can hear you clearly, sir. Some noise disturbance is there. கொஞ்சம் விட்டு விட்டு வருது சார். பிரேக் டவுன் ஆகுது, வாய்ஸ் பிரேக் டவுன் ஆகுது சார்.
ஒரு நிமிஷம்.
வாய்ஸ் பிரேக் டவுன் ஆகுது சார். வாய்ஸ் பிரேக் ஆகி கிடைக்கும்.
ஹலோ ஹலோ சார் மறுபடியும் பிரசன்ட் பண்ணுவோம் சார். பேக் போயிட்டு ஹலோ ஹலோ
சார் கேக்குது சார். இப்ப கேக்குது சார். So we can hear you.
Okay.
Sir, you can proceed, sir. Yes, sir. Please, sir. Proceed, sir. Proceed. We can hear you now.
Inhibit the causes the which thickens the entire epithelium and leads to cervical cancer. Here, this is the overall view of how the normal cervix and the cancer stage cervix which show. Here, what are the mechanism of E6, E7 in carcinogenesis in cervical cancer? HPV infection, I already told that E6, which downregulates the lead transformation and inhibition of the women worldwide. Radiotherapy treatment, which is the main modality to treat the all stages of cervical cancer. It plays a major role in radio resistance and chemo. So hence we hypothesized that fustos homolog can be a target of fustos homolog. What is fustos homolog? Initially, FTS, which is identified in the mouse mutant, due to the defects in the limb development, they observed the limb development and they referred as FT1 and FTS. And they found several pairs of the deletion of mutations and which occurred in of interaction with the AKT, which is a cell survival marker, control the AKT by the PDK1. FTS in human cancer. The investigated the role of FTS in numerous cellular signaling, such as and other chemotherapy drug EGCG treatment. Here, for the first, they speculated FTS may involved in the radio resistance based on the DNA microarray by our previous lab member, Dr. Senthil Kumar. There are they find some set of says that FTS overexpressed FTS and seen that the phosphorylation of EGFR is increased and the colony formation is reduced in cells. Researcher shown that epithelial mesenchymal transition were greatly reduced by FTS silencing. Identified the expression pattern of FTS for the first in human cervical cancer. and century to carcinoma, we see abundant expression of FTS, which by FTS. Here, what are the relationship between FTS and EGFR signaling and radiation sensitivity? Overexpression of EGF receptor is a well-known receptor related to chemo and radio resistance and a poor prognosis in many cancers. EGFR, which is activated by the chemotherapeutic drugs cisplatin and radiation, and this activation which leads to the resistance to the chemotherapy as well as the radiotherapy. Protein kinase, which is a protein involved in the DNA repair. And here we delineates the role of regulatory role of FTS in EGFR signaling. Here the picture shows the how EGFR signaling works. Cell proliferation, invasion, metastasis, and inhibition of apoptosis. Here there is a and in turn translocate into the nucleus, where the radiation-induced damage click for which are gefitinib, erlotinib, and other kind of antibodies such as cetuximab, panitumumab. Here we wanted to see whether FT silencing have any effect in this signaling path. First experiment. First, we irradiated the cells of ME180 cervical cancer cell. And in both cytoplasm as well as in the nucleus, which suggests that and change is the experiment we chose and EGFR in the nucleus. In silent cells, the translocation of EGF receptor is completely reduced. And then the receptor of FTS with EGFR after 4-grade radiation treated. And then we wanted to address the functional consequence of FTS and EGFR. Here increase in the 4-grade radiator treated group of EGF receptor and phospho-EGFR and FTS. Here in the regulation of EGF receptor and phospho-EGFR. And then we wanted to check the gamma HTX, which is a surrogate marker to study the double-stranded breaks induced by the radiation. Hence, we chose the 2 time points late 24-hour. And the 24-hour time point. But if you seen in the FTS silence treated group, it here can see the abundant expression of gamma H2X marker by radiation, which is again to check the physical association between result which concludes that FTS and EGFR, which are physically associated, FTS would be the therapeutic target to increase the radio sensitivity in cervical cancer. Effect of silencing in cisplatin sensitivity by inhibiting the EGF receptor mediated repair of DNA damage. Here we speculated the cisplatin treatment overexpress the EGF receptor, which leads veins. Here, these are the materials methods which we used. Move on to the first experiment. We done a 0 to 50 micromolar concentration of cisplatin drug. We observed a dose-dependent reduction of cell numbers in cisplatin treated group, but the SIGA cells, which are the less toxic, least sensitivity among the other three cell lines. And here we observed a IC50 value of 11.5 micromolar of cisplatin. Then we chose a lower dose to check the pattern of EGF receptor and phospho-EGF receptor. Here we chose 0.1, 1 and 10 micromolar concentration of cisplatin. Here cisplatin increases the EGF phosphorylation EGF receptor and if we see in the other cell lines, we are not able to see the increased amount, but these cells which are sensitive to the cisplatin. And then we wanted to assess the subcellular localization of EGFR and phospho-EGFR in the cytoplasmic and then we want to assess what are the changes occurred by FTS silencing. Here, the increased expression of EGFR and phospho-EGFR by and then we wanted to investigate the FTS silencing augment cell death by cisplatin. Here, the photomicrographic image which shown the cisplatin treatment alone, and the next one here we can see a increased amount of cell death compared with cisplatin treated alone in FTS silenced combined cisplatin group. Here we performed with the two cell lines HeLa and MU-80, and then we wanted to determine the expression of DNA PK, which is 10 micromolar treated group, gamma XTX, which found least amount of expression, but in FTS silenced group, the amount were doubled, which shows FTS silenced combined cisplatin increases the double standard rate. Here we perform the immunofluorescence assay to see the expression pattern of gamma XTX. Me180 cells. And then here we shown the amount of phospho-DNA PK which is increased in cisplatin treated group, and FTS prevented the reduction of phospho-DNA PK. We wanted to assess the physical GFR and DNA PK, but the least amount is which shows EGFR mediated repair of DNA damage induced by cisplatin. FTS is taken together. We propose that the increased efficacy of cisplatin in cervical cancer cells FTS in notch. Here, notch signaling, which is a well-known signaling path, ligand activation, the notch which occurred in a series of cleavage, proteolytic cleavage occurred, and the activated form of notch 1, that is called to be a notch intracellular domain, which translocate to the nucleus. Target in histone proliferation and the stem cell maintenance, the overall which leads to cancer. Also, the signaling which is involved in the other cell proliferation possible relationship between FTS and signaling.
Sir, excuse me.
Including cervical cancer. Hello.
Sir,
Also, the
maybe network
signaling
issue
which is
side
involved in
voice
the
breakdown
other cell proliferation. Hello. Sir, can you hear me?
Yes, I can hear you, sir. But உங்க சைடு ஏதோ நெட்வொர்க் இஷ்யூஸ் இருக்குன்னு நினைக்கிறேன் சார். கொஞ்சம் இடையில பிரேக் ஆகி தான் வருது சார். ஃபுல்லா கம்ப்ளீட் ஆக மாட்டேங்குது.
Oh, possible relationship between FTS and signaling.
Sir, excuse me.
Including cervical cancer. Hello.
Sir,
Also, the
maybe network
signaling
issue
which is
side
involved in
இருக்குன்னு வாய்ஸ்
the
பிரேக்
other
டவுன்
cell
ஆகுது.
proliferation. Hello.
சார் கேக்குது சார், சொல்லுங்க சார்.
பிரசன்ட் பண்ணலாம்.
பண்ணுங்க சார், பண்ணுங்க.
இல்ல டேய்
சார் பண்ணுங்க சார், பண்ணுங்க.
ஓகே ஓகே சார்.
இடையில ஃபுல்லா வரல, இடையில கட் ஆயிருது. பேசிட்டே இருக்கிறப்ப கட் ஆகி ஒரு நிமிஷம், ஒரு செகண்ட் 2 செகண்ட்ஸ் கழிச்சு திருப்பி வாய்ஸ் இதாகுது. மேபி உங்க நெட்வொர்க் கனெக்டிவிட்டில எதுவும் இஷ்யூஸ் இருக்குன்னு நினைக்கிறேன் சார். இப்பவே நீங்க பேசுறது ஸ்டாப் ஆயிடுச்சு சார். ஸ்டாப் ஆகி ப்ளே ஆகுது, ஸ்டாப் ஆகி ப்ளே ஆகுது சார்.
ஓகே, கண்டினியூட்டியா இல்லையா?
ஆமா சார், ஆமா சார். கட் ஆகி வருது. இந்த நெட்வொர்க் இஷ்யூஸ் இருக்கு இல்லையா மொபைலில் அந்த மாதிரி இருக்கு சார். இப்பவுமே ஹோல்ட் ஆயிடுச்சு.
ப்ரொசீட் பண்ணவா சார்? ஹலோ
பண்ணுங்க சார், பண்ணுங்க. உங்கள்கிட்ட ஒரு இன்டிமேஷன் தான் சார். உங்க சைடில் எதுவும் நெட்வொர்க் எதுவும் வைஃபைல இருந்தீங்கன்னா வைஃபைல எதுவும் கனெக்டிவிட்டி எதுவும் ப்ராப்ளம் இருக்கும், அதுக்காக ஒரு இன்ஃபர்மேஷனுக்காக தான் கூப்பிட்டேன், மற்றபடி ஒன்றும் இல்லை. ஒன்றும் இஷ்யூ இல்லை சார். அது ஸ்டாப் ஆகி ப்ளே ஆகிற மாதிரி இருக்கு. வாய்ஸ் கட் ஆகி திருப்பி பிரேக் ஆகி திருப்பி ஒரு செகண்ட் கழிச்சு திருப்பி வருது. ஒரு சின்ன இன்டரப்ஷன் மட்டும் இருக்கு.
ஓகே சார்.
ஓகே சார், தேங்க்யூ சார். நீங்கள் ப்ரொசீட் பண்ணுங்க சார். வைஃபைனா கொஞ்சம் லைட்டாக கொஞ்சம் மூவ் பண்ணி வைஃபை சிக்னல் கரெக்ட் ஆகிற மாதிரி கொஞ்சம் மாற்றிக்கோங்க, அதுக்காக சொல்லுங்கள் சார். தேங்க்யூ சார், தேங்க்யூ.
Here, ionizing radiation which activates the notch signaling pathway, which resulted in the increased number of cancer stem the radio resistance. In this study, we identified the regulatory role of FTS in radiation-induced notch-on Here, these are the materials and method which we used to prove our hypothesis. In the, notch, that is the cleaved notch one, after 4-gray irradiation treatment. We increased time dependent of notch and the notch downstream targets in ME180 cells. But in the other cells, we are not. And then we wanted to check whether the FTS silencing have any effect in this notch signaling. In the previous slide, we by radiation finding where we see the co-localization of FTS and notch1 in the human cervical tissue sample. To check the interaction screen between the notch1 and FTS, prevented the nuclear another immunofluorescence assay to see the co-localization of the notch active form, which is the cleaved notch1. Here we observed induced cleaved notch1 expression and FTS after cleaved notch1 in the nuclear region. And then the silencing of FTS, which prevented the nuclear notch1 as well as the notch target S1 in gamma secretase complex marker of notch intracellular domain to this all set of four proteins which are increased by irradiation and silencing of FTS reduces its nuclear import in this ME180 cervical cell. Shocking confirmation of notch and FTS by using a in silico model. Here, green color and the pink color, which is the notch, we see a clear interaction by using in silico model. And then we performed the immunoprecipitation assay to see the functional 4-gray of radiation. Move on to the next slide. Here the tube the tumor spheroids which are grown in the ultra-low attachment plates with supplement of EGFR, epidermal 20. You can see radiation affects the spheroid formation ability, but here foreground irradiation does not. Completely disrupt the these are the three set of markers which are involved in the pluripotent. Here we checked the protein expression of Notch1 in cells steroids. Expression of Notch signaling molecules, and FTN silencing completely reduces all the Notch signaling molecule and here we performed the immunofluorescence assay. Here, and in Western blot expression, in cervical cancer. The overall result which suggests that radiation increases the protein expression of overall, which includes FTS, which is a potential target for Notch-mediated radio resistance in cervical cancer. And here, this is the and reduces the spheroid forming ability and target the stem cell markers. Here, I want set of oncoprotein E6 and E7 which are responsible for cervical cancer progression. Both the set of E6 and E7, as well as it increases the tumor suppressor protein P53. I thank the institution for giving me the opportunity. And please open the question, sir. Thank you.
Thank you, sir. Thank you, sir. Thank you for your wonderful presentation, sir. This forum is open for questions. Sir, can you tell me about that fused dose homolog, sir? A little bit elaborate, please, sir.
Yeah. Hello.
Yes, sir. Yes, sir. I can hear you, sir.
Sir, initially, no. Set of genes were upregulated by radiation. So, in the first in different time points. Just a minute, sir. Hello,
Yes, sir. Yes, sir.
sir, can you hear me?
Yes, sir.
Hello.
Yes, sir.
The previously, several researchers shown the increase. Hello. Hello. Hello.
Sir,
Hello.
sir, please.
சார் கேக்குதா சார்?
சொல்லுங்க சார்.
சார் ஸ்லைடு தெரியுதா?
தெரியுது சார். ஸ்லைடு தெரியுது சார். சார் வணக்கம் சார். கட் ஆயிடுச்சு சார் நீங்க.
Hello.
Yes, sir. Please, sir.
Yes, sir. ஸ்லைட்ஸ் தெரியுதா சார்?
ஸ்லைட்ஸ் இன்னும் ஷேர் பண்ணல சார் நீங்க.
இதோ ஒரு நிமிஷம்.
Yes sir, it is visible sir.
வாய்ஸ் ஆடிபிளா இருக்கா சார்?
வாய்ஸ் ஆடிபிளா இருக்கு சார். பிரேக் டவுன் எதுவும் இல்லாம இருக்கு சார்.
ஓகே சார்.
So the question is
சொல்லுங்க
sir in cervical carcinogenesis.
Sir, initially our previous lab members checked the DNA microarray. What are the set of genes which are upregulated after irradiation? They found and one among that they found FTS. And then they thought FTS may be involved in the cervical cancer progress. But here, from SYN1 to SYN3, we can see the gradual increase of FTS, which shows FTS might be involved in the progression of cervical cancer. What the is FTS oncogene which driven the cervical cancer progression, sir? Hello. 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 Sir. Hello, sir.
Hello.
Yes, sir.
Yes sir. Can you hear me sir?
Yes, sir. Yes, sir.
Yes sir. Yes sir. So what is the role of HPV in the diagnosis or the treatment, sir? Like you have told, HPV is FSD, fused dose homolog is a gene expression, a protein which is present in the HPV, sir, in the presence of HPV. So what is the role of this fused dose homolog in diagnosis or treatment of cervical cancer, sir?
So here we strongly believe that absence of FTS increases the radiosensitivity. We've shown the increased amount of apoptosis in HeLa cells, sir.
So then how will you extract this FDS, sir, from the cervix, sir?
Sir, by using silencing RNA. Hello?
I can hear you, sir. I can hear you, sir.
Yes, yes. Here I'm showing the unpublished results. Can you see this slide? Sir?
I can see you, sir.
Yes, sir. In this unpublished finding, we elaborated the FTS with E6 and E7. in the presence of FTS, we can see the increased expression of but both the oncogenes, set of oncogenes, which means FTS is required for the HPV-mediated E6-E7 progression in cervical cancer. So this is a oncogene which driven E6-E7 mediated cervical carcinogenesis.
Okay, sir. Any other questions? Any other questions? Sir, thanks, sir.
Thank you, sir.
Sir, thank you, sir, for spending your valuable time, sir. We have some little bit network issues, sir.
I'm sorry.
Yeah, we can. Yes, sir.
Yes.
We can get you clearly, sir. Sometimes there is a broken voice was there. But I think hopefully it may help our students if they want to pursue their career in the research, sir. So thank you for spending your valuable time, sir.
Yes, sir. Yes, sir.
Sir, and one more request, sir. If any of your friends are interested to share your research experience in our international webinar series, kindly refer to us, sir.
Sure, sir. Surely I will let you know. Yes.
And definitely, sir. And whenever you are coming to Tamil Nadu, kindly let us know, sir. We can invite you to our institution. You can have a direct interaction, and you can share your research inputs with our students.
Sure. In college, if you want to do some collaboration work or something, please let me know, sir, regarding MOU signing with the Korean.
Definitely, sir. Definitely, sir. I have saw your profile, sir, and you have been a part of that Korean Tamil Sangam.
Yes.
Yes, sir. I think you are doing some lot of works in Korean Tamil Sangam. You have been editorial board also, sir. So definitely help our students. If anybody interested to join higher studies in Korea, also we will get you back, sir.
Sure. Surely I will help, sir.
Okay, sir. Definitely, sir. So thank you, sir. Yes, sir. Please, sir.
And one more thing. Last week, institution website.
Yes, sir.
You have conducted more than hundreds of webinars.
Yes, sir.
Yes.
By our principal's effort, sir. Our principal is doing exercises involving all these activities, sir. And
yes, the institution is working round the clock for the student welfare.
Yes, sir.
Yes, my hearty congratulations to you, to the team of staff members.
Okay, sir. Thank you, sir. Thank you very much.
I think the students are to have teachers like you, sir.
Definitely, sir.
Thank you for your hard work, sir. Thank you.
Thank you, sir. Thank you very much, sir. Thank you very much, sir. So we will let you know, sir. Any other future help, we will mutual research work. We will get you know, sir. Just help us, kindly help us.
Thank you.
Thank you, sir. So thank you for spending your valuable time with, sir.
Thank you.
Okay, thank you, sir. Thank you very much, sir. Take care, sir. Bye, sir. Bye.